Good evening to all. Uh, Myself Jyoti Chaitanya. Uh, in the last meeting, we have discussed about the introduction about the proteins, and we also discussed about the structure of the proteins. And today, I wanted to discuss you about the classification. One second. Uh, about the classification and characteristics of the protein. Here, as we know, the proteins which containing the carbon, hydrogen, oxygen, nitrogen, and sulfur serves as structural components of the animals. And here you can see the structure of the proteins, which is composed of a chain of amino acids. Here as per our last class, uh, we know there are 20 possible groups of the protein. Here, the amino acids, which are also called as the building units of the proteins, and the proteins are the polymers of amino acids, which link together by called peptide bond. There are 300 amino acids occur in the nature. Only 20 of them are in proteins. And when coming to the structure of the amino acids, here you can see the structure of the amino acids, which is having the amino group and also having the carboxylic side chain. Here you can see the peptide bond, which forms form in between the two amino groups, amino acid 1 and amino acid 2. Here you can see the peptide bond, polypeptide chain when the condensation happens. When coming to the classification of the proteins, here the classification of the proteins, which is depending upon the composition of the proteins and also depending upon the hydrolysis of the products. Here, depending upon the composition, here we have the fibrous proteins and globular proteins. Here the fibrous proteins are the keratin, collagen, myosin and fibrin, whereas the globular protein called hemoglobin, insulin. And depending upon the hydrolysis, here we have the simple proteins, conjugated proteins and derived proteins. Here the simple proteins are albumins and globulins, whereas conjugated proteins are again classified into nucleoproteins called nuclein and glycoproteins called mycin and chromoproteins called hemoglobin and chlorophyll. And the other one is the derived proteins. Here you can see the proteoses and peptones and peptides. When coming to the fibrous proteins and globular proteins, here the fibrous proteins, which is consisting of long parallel polypeptide chains, which forming the helical structures or plated sheets, and globular proteins, which consists of a coiled and folded polypeptide chains, which forming the spherical shape, which is solubilized in the water, whereas the fibrous protein, those were insoluble in water. And the structures are stable in case of fibrous proteins, whereas the structures is unstable in case of globular proteins. And a fibrous protein which plays a major role in mechanical and structural function, whereas the globular proteins that takes part in metabolite and chemical processes. Here the example for the fibrous proteins are keratin and collagen, and the enzymes and hemoglobin are the example for the globular protein. Here you can see the structure of globular protein in case of hemoglobin and myoglobins are examples of globular protein. And fibrous proteins are scleroproteins. Those were presented in the collagen and keratin, elastin. Those were the examples for the fibrous proteins. And the classification which is depending upon the composition of the proteins are simple proteins, conjugated proteins, and derived proteins. Single proteins, they are hydrolyzed to amino acid residues. When the proteins are hydrolyzed, it produces the simple proteins. Those were histone and albumin. When coming to the conjugated proteins, name itself indicating that there will be addition to the protein group. What is that? Here it is the non-protein group. Here the non-protein group, we consider it as a prosthetic group in their structure. The example for this one is neuroprotein, chemoprotein, and nucleoprotein. When coming to the derived proteins, they are formed from the native proteins by adding heat. Those were the primary derived proteins, for example, metaprotein and proteins. And the secondary derived proteins are peptides and peptones. And when coming to the conjugated proteins, as we already discussed in the last two papers, those were nucleoproteins, glycoproteins, mucoproteins, lipoproteins, and phosphoroproteins, and chromoproteins, metalloproteins. Here you can see the structure of glycoproteins, which is presented in the follicle stimulating hormones and luteinizing hormones. And here you can see the structure of lipoproteins. Those containing the cholesterol esters, cholesterol, triglycerides, and phospholipids. And classification of proteins depending upon their function. We already discussed all these in the last class. Structural proteins, protein examples like keratin, collagen, and elastin. And the structural organization of the protein also we discussed in the last class. So here the other topic is complete and versus incomplete proteins. Here the complete proteins which the dietary protein is required for the body, as there are 9 essential amino acids in the body that cannot create and must obtain from one's diet. A complete protein which contain all 9 of these essential amino acids, that versus incomplete proteins which do not. The complementary proteins are combining of the 2 or more incomplete proteins that supply 9 essential amino acids. When coming to the complete protein, which is animal-based one, meat, poultry, dairy, eggs, and fish. These were the examples for the complete proteins. In case of incomplete proteins, plant-based things like vegetables, grains, legumes, beans, nuts, and seeds. These were the examples for the incomplete protein. When coming to the complementary proteins, grains, legumes, vegetables, nuts, seeds are the examples for the complementary proteins. And what are the important functions of the proteins? Yes, definitely. Proteins plays the major role for the structural components. So that, as we know, the collagen is in tendons and cartilage, keratin in hair, skin, wool, and nails. Those were plays the major role for the providing the structural components. And movements of the muscles, that is contractile movements, myosin and actin are the contractile muscle fibers. Transport, carry essential substances throughout the body. Those were hemoglobin, which transport the oxygen, lipoproteins transport the lipids. And storage, casein, which stores the protein in milk, and ferritin, which stores the iron in the spleen and liver. When coming to the hormones which regulate the body metabolism and the nervous system, as everyone knowing that insulin that regulates the blood glucose level, and the growth hormone which regulates the body growth, and enzymes which catalyzes the biochemical reaction in the cells, the sucrase, which is the enzyme that catalyzes the hydrolysis of the sucrose, and trypsin is the enzyme that catalyzes the hydrolysis of the protein, and protein protection. Yes, we know the when the body is attacked by the foreign substances, that can be recognized and destroyed by the immunoglobulins. Those were the proteins which stimulate the immune response. These were the important functions of the protein. And when coming to the biological significance of the proteins, all the enzymes are the proteins, as we know, and which is storing the amino acids as nutrients, and which acts as the biological building blocks for the growing organism, and which transport the functions like protein transport fatty acids, bilirubin, ions, and hormones, some drugs. Proteins are essential elements in contractile and motile systems like actin, myosin, and which protective and defensive function like fibrinogen and antibodies. Some hormones are proteins, as we know, insulin, somatotropin, and some structural functions like collagen and elastin. So, our next topic is this is all about carbohydrates and the proteins, and amino acids also we already discussed in our last topic. Hello? So the next topic is the digestion. Why? Because we already discussed about the biochemistry of the proteins and carbohydrates. Here we can see the transamination and deamination. Here you can see. ఒక్క నిమిషంలో ట్రై చేస్తాను. దీంట్లో దాంట్లో మనసు దాంట్లో ఇప్పుడు ఉందా మన ఇంటి లొకేషన్ పెడతా. నేనే బుక్ చేస్తాను, ఒక్క నిమిషం. Here you can see the transamination and deamination. As a part of metabolism, in animals, after protein is digested, the amino acids are absorbed into blood circulatory system. Once it reaches to the liver and other tissues, in the liver, amino acids can be utilized for the energy production. In the process of amino acid which break down the ammonia that formed from the amino group is converted to the urea, and that will be excreted from the body. Besides, the amino acids are also utilized for the protein synthesis and other biologically important substances. What is transamination? Name itself indicating that something is removed. What is something here is amino group. Here, in the first step, in the catabolizing, this is also called as catabolizing reaction or breaking down. What is breaking down here? A complex molecule is breaking down into a simple molecule. Here the removal of amino group is taken place. After the amino group is removed through the transamination or the deamination respectively, the transamination is the transfer of an amino group from the amino acid to the keto group. Amino acid without an amino group, thus creating a new amino acid and the keto acid. This reaction is catalyzed by a family of the enzymes called as transaminases. Transaminases are the enzymes which remove the amino group from the protein. So what actually happens here is the the transamination reaction, the result of this one is exchange of amino group on one acid with a ketone group on another acid. It is analogous to the double replacement reaction. The most usual and major keto acid that involved in the transamination reactions is alpha ketogluteric acid. And the intermediate, which is the intermediate compound in the citric acid cycle. A specific example for the transamination of the alanine to make a pyruvic acid and glutamic acid. Other amino acids which can be converted after several steps through the transamination into the pyruvic acid, that includes serine, cysteine and glycine. In this reaction, there is no ammonia involved, and that is catalyzed by the specific transaminations which are believed to be produced into the muscle tissue. Yesterday I tried to open my screen, but here there will be a problem with my computer, I guess. So I'm trying to make it possible, but still I'm unable to doing that. Sorry, Minhas. Though I'm telling the class, but still I'm trying to rectifying the problem. It's not working on. I don't know why.
It's okay, ma'am. No problem.
Thank you. Shall I continue with the class?
Yes.
Okay. Something is opening. Okay. Here you can see the structure of transamination. How transamination happening here, the removal of amino group. When coming to the here you can see from the amino acids that gives rise to the acetyl-CoA and are said to be the ketogenic. Certain amino groups are fall into the both categories. Those were leucine and the amino acid ketogenic group. And when coming to the deamination, here the deamination, whenever the excess amount of the proteins are presented in the body that are converted into the carbohydrates, or the fatty acids. Here, that fat that are used for the energy or storing. So, but proteins which containing the nitrogen group, which is removed by a process called as deamination. Here, what is happening in case of transamination? The removal of amino group is taken place. But here, in case of deamination, name itself indicating that here the proteins which containing the nitrogen group, which is removed by a process called deamination. Before amino acids are utilized as carbohydrates or the protein. Here, whenever the excess amount of the proteins are presented in the body, then only the deamination reaction happens. So, this is the process which is catalyzed by the specific amino acid oxidizes and lead to the formation of the corresponding alpha ketogluteric acid and ammonia. These enzymes are presented in liver and kidneys. Deamination is also an oxidative reaction that occurs during the aerobic conditions in all tissues, but especially in the liver. During the oxidative deamination, an amino acid which is converted into the corresponding keto acid by the removal of the amine functional group as ammonia, and the amine functional group is replaced by the ketone group. The ammonia which is eventually goes into the urea cycle. Oxidative deamination that affects the replacement of the amino group by the oxygen. And this reaction, which is catalyzed by the amino acids called amino acids oxidases, an enzyme which containing the oxidizing coenzyme A, that is fat. The reduced coenzyme is reoxidized by molecular oxygen to form the hydrogen peroxide, H2O2. And oxidative deamination occurs primarily on glutamic acid because glutamic acid was the end product of many transamination reactions. The glutamate dehydrogenase is the allosterically controlled by the ATP and ADP. ATP acts as an inhibitor, whereas ADP is an activator. Adenosine triphosphate, which acts as an inhibitor, whereas ADP, adenosine diphosphate, is activated acts as an activator. Here you can see the overall reaction. The removal of nitrogen group may take place from the amino acid and forms into the keto acid and ammonia. Here it is a ammonia. So A missed here. So here the keto acid that which enters either the carbohydrate or the fatty acid cycle, which is produced from the above reaction. The keto acid again enters into the carbohydrate or fatty acid cycle for the completion of their degradation process. Furthermore, ammonia formed by the deamination of the amino acid in the liver, which is converted into the urea. Yes, that urea is excreted through the urine. Here, the leucine, isoleucine, phenylalanine, and tyrosine are the ketogenic because they are converted to the ketone bodies, acetoacetic acid. Threonine, valine, and many other amino acids are the glycogenic and gluconeogenic, since they gives rise to the compounds that can readily be converted to the glucose. Apparently, most of the amino acids may be deaminated, but this is a significant reaction only for glutamic acid. Other amino acids are deaminated with the combination of the transamination and deamination of the glutamic acid. which occurs, which is a recycling type of reaction for the glutamic acid, where the original amino acid that loses its amino group during the process. When the process is happening in between, the whichever the original amino acid here we have, that loses its amino group. That amino group is formed into the ammonia. Here you can see oxidative deamination and glutamic acid oxidative deamination reaction. So here ammonia, which is as everyone knowing that, which is toxic to the cells, and urea, non-toxic to the cells, which is excreted from the urine. Here you can see in the picture which representing the deamination reaction. So there are some specific amino acids called oxidases that require the pyridine nucleotides. The reduced coenzymes called NADH and NADPH are oxidized by the way of respiratory chain. The most important specific oxidases is a glutamic acid oxidases. The reverse of this reaction that shows a means for the synthesis of the amino acid from ammonia, which is an intermediate of the carbohydrate metabolism. The amino group is the glutamic acid that can be passed through transamination reaction that producing all the other cellular amino acids that provides the appropriate alpha ketogluteric acids that were available. Here you can see the interrelationship between the amino acid pool and the metabolic pool. Here the glutamic acid from the formation of the alpha ketogluteric keto group from the glutamic acid. The same glutamic acid here you can see in the metabolic group. When coming to the urea cycle or ornithine cycle, here the utilization of amino acids for energy or synthesis of glycogen or fatty acid, an amino group which is deliberately in the form of ammonia. In the last class we discussed when as we know, ammonia is toxic to the human body. How much toxic it is? If it is presented 5 mg in a 100 ml of blood group, it is becomes like a poison which retains in the body. So the normal concentration of this is about 1 to 3 micrograms in the 100 ml, which is excreted through the kidneys as a urine. Under certain pathological conditions, a large amount of the uric acid cycle are produced and deposited as the soluble monosodium salt. Those were deposited, which is occur in the joints and produce the painful symptoms characteristics of the gout. Most of the people, once the people reach to the age of 30 and 35, in the soon after they wake up from the bed, they used to feel like pain in the joints. This is happened only because of the consumption of less amount of the water. If the water level is more, that flushes the ammonia from the body. If not, definitely that leads to the pains in their joints. As well as some people, while they are sitting and while they are walking, they will feel like some pop sound from their joint region because of the presence of H2O2, hydrogen peroxide over there. Because extensive amount of the ammonia leads to giving some sort of pop sound and extensive amount of H2O2 also. Because of that reason, people used to A doctor used to recommend the people go for walking and doing some exercises. So, for making the metabolism properly. So, the principal here, the involvement of the principal organ, which is concerned in the uric acid cycle, is liver. Extensively, liver may get damaged because of the accumulation of high amount of the ammonia. Definitely, that may lead to the death. Once the urea is produced, that is transported by the bloodstream to the kidneys and which is eliminated in the urine. Normal individual that excretes about 25 grams of the urea daily, although this value which varies greatly from day to day and is depending upon the protein intake. The synthesis of the urea, uh, what we call the synthesis of the urea is called as urea cycle or ornithine cycle. What happens in the first step of synthesis of urea? Here the formation of the carbamyl phosphate from the ammonia and CO2 in the as a initial step in the urea synthesis. Here you can see the formation of the carbamyl phosphate. The involvement of the reactants here are the ammonia, carbon dioxide, and two molecules of energy. Here, the energy for the process which is supplied by the two molecules of the ATP, the mechanism is not being cleared. So, the next step is the high energy compound, the carbamyl phosphate, then reacts with the ornithine in the presence of the ornithine carbamylase to form the citrulline. So here the formation of the citrulline taken place. Here you can see the structure of the ornithine. Ornithine carbamylase is an enzyme that converts the ornithine into the citrulline along with an inorganic phosphate is released. The next level is the conversion of citrulline to the arginine, where the involvement of the two enzymatic reactions taken place. The first one is the reversible condensation of the citrulline with the aspartic acid in the presence of arginine succinic synthetase. The reaction which produces the arginosuccinic acid, which is a stable intermediate compound that cleaves in a subsequent step to yield a molecule of arginine and a molecule of fumaric acid. The synthesis of fumaric acid is important since it can be converted through the Krebs cycle to form the OAA, oxaloacetic acid. That latter that can be transaminated means the removal of the amino group taken place for yielding the aspartic acid. So here with the acetyl-CoA group that forms into the citric acid that you can see here. In the final step where the arginine, here you can see the arginine, that arginine is hydrolytically cleaves by the arginase that yielding the urea and regenerating the ornithine. The latter is now ready to accept the another molecule of the carbamyl phosphate. This is how the cycle happens. One of the nitrogen atom that is eventually converted to the urea is contributed by the aspartic acid, that not by the ammonia. However, the ammonia that may be incorporated into the aspartic acid. This is how the entire the urea formation cycle happens. What happens here? The ammonia and here the reactants are the ammonia, carbon dioxide and water molecule. Here the three energy molecules called 3-ATP along with aspartic acid that forms into the urea and fumaric acid along with the releasing of two energy molecules, 2 ADP and adenosine monophosphate along with one inorganic phosphate and one one PPI. Here, the dietary proteins, when during the digestion, that releases the amino acids and glucose, galactose, glycerol, and fatty acid. Here, the amino acids that enter into the nitrogen pool, whereas glucose, fructose, galactose that enter into the blood sugar and glycogen pool. So, here the glycerol fatty acids that deposit the fat and plasma lipid, where the nitrogen pool, the conversion of the urea taken place that we already discussed in the last cycle, that is urea formation, and it is also utilized for the tissue and protein formation. This is also utilized for the oxidative deamination and transamination reactions. Those were enter into the glycolysis as a pyruvic acid that converted into the acetyl CoA. This is here the nitrogen is converted into the pyruvic acid as well as acetyl CoA. Whichever the pyruvic acid releases, that is enter into the acetyl coenzyme cycle. There, acetyl CoA is directly enter into the citric acid cycle where the energy is generated. This is how the interrelation of the metabolic pathways taken place. Here the deposited fat and plasma lipids, those were converted into the lactic acid. That lactic acid again converted into the pyruvic acid. Along with that, during the fatty acid synthesis and the lipogenesis, these were converted into the cholesterol. These were converted into the acetyl CoA. Here the releasing of cholesterol and the bile salts may be taken place. This is how the interrelation in between the metabolic pathways happens. When coming to the summary, we already discussed about what is protein here. Yes, protein means the long chain of amino acids. Proteins are the essential constituents of the cells. These are the constituents which may be presented in the animal body. They account for the about one half of the dry weight of a body. Here, the amino acids which is linked to the peptide bond, both of the amino acids which is linked to the peptide bond, amino group and carboxylic group are called as the proteins, which is also called as amphoteric in nature. And the animal resources sources of proteins are the milk, egg, meat, fish, liver. Whereas in case of plants, pulses, nuts, and cereals. Depending upon the molecular structure and depending upon their chemical composition, solubility, proteins are classified. And here the when the amino acids are broken, when the protein is broken down, the amino acids, when the amino acids are removed, amine group is removed, that reaction is called as transamination or deamination. Here the transamination means transfer of amino group from amino acid to the keto acid. So the amino acid which is without the amino group is called as transamination. When the excess amount of the proteins are presented in the animal body, that is converted into the carbohydrates and the fats that are used for the energy or storing. But the proteins which containing the amino group, which is removed by the process called deamination, before amino acids are utilized as a carbohydrates are the proteins. Here, the oxidative deamination that affects the replacement of the amino group by the oxygen. This reaction is catalyzed by the amino acids oxidases, which is the enzyme that containing the oxidizing cofactor, that is FAD, which utilization of the amino acid for the energy or synthesis of the glycogen or the fat. An amino group which is liberated in the form of amino, which is the poisonous material which retains in the body. The synthesis of urea is also called as urea cycle or ornithine cycle. So this urea is flushed out from the body through the kidneys and liver. This is about the proteins. When coming to the lipids, yes, lipids, as everyone knowing, uh, lipids are the heterogeneous group of the organic compounds that are important constituents of the plants and the animal tissues. Yes, uh, they are the group of biological molecules. Their solubility in the organic solvents such as benzene, ether, chloroform, and carbon tetrachloride, CCl4 అంటాం కదా, that one. And their insolubility in water, definitely most of the fats are insoluble in water. There will be a presence of edible lipids that constitute approximately 25% to the 28% of the diet. And lipids that was stored in the body, almost 90% as like a fat, which is stored in the body, where the fat deposits are known as subcutaneous tissues, intermuscular, perinephric, omental, and mesenteric tissues that stores a neutral fat in their adipose tissue, which may contain 6,000 grams of fat in the 70 kg of the human body. Brain and the nervous system in the large amount, and all other tissues in the smaller amount, which containing the phospholipids, glycolipids, cholesterol, and other total reaching about 3,000 grams. So when coming to the liver containing the 20 grams of neutral fat, and 50 grams of phospholipid and 5 grams of cholesterol. The blood and the body fluid, which containing the 75 grams of the different lipids, thus 10 kg of lipids in the 70 kg weight humans. If the human is having the 70 kg of weight, definitely they'll have the 10 kgs of lipids, means as a cholesterol. Here the lipids are an important dietary constituents. They include fats, waxes. As we know, presence of fats is very dangerous to the body, but still, definitely, it is the important dietary constituent which uh, uh, which uh, which is as like carbohydrates and amino acids which we require for our metabolism. And here the name lipid, which is given to the heterogeneous compounds because of their insoluble nature in the water. But generally, they dissolve in the organic substances like chloroform, the carbon tetrachloride, ether, and alcohol. Lipids that have the elements called carbon, nitrogen, and oxygen as like the carbohydrates. In addition to the carbon, hydrogen, and oxygen, it is also having the phosphorus and nitrogen in it. Since the carbon and hydrogen ratio to the oxygen in the lipids, it is higher than the carbohydrates. The energy yield is more than the carbohydrates, like per gram in the lipids. Here you can see the fats 9.3 calories of uh, energy, whereas proteins that will show 5.3 calories, whereas carbohydrates that will show 4.1. So definitely it is more than the proteins and carbohydrates. So as we know, what are the sources of lipids are? Yes, for the animal source, milk, cheese, butter, eggs, meat, fish. While in case of plant fats, a mustard oil, a sunflower, groundnut oil, gingerly oil, vanaspati, and there are many other sources that we regularly use in the food, use as a food and use in the food preparations. So when coming to the classification, here as just we discussed, these are the esters of fatty acids. So here we have the simple lipids, saturated fatty acids, and um, here we have the complex and compound lipids. As just we discussed, it is having the phosphorus and sulfur. Based upon that, here we classified the lipids into complex and compound lipids. And the other one is derived lipids, how the lipids are formed, how the lipids is classified through as a derived lipids. Here, coming to the simple lipids, these are the esters of fatty acids with the glycerol. Here, three fatty acids joined together forms into the a simple lipid called triglyceride. Upon the hydrolysis, um, this yields the fatty acid. Depending upon the presence of double bond in its molecular structure, fatty acids again classified into saturated and unsaturated fatty acid. Here the unsaturated fatty acids that possesses the bond between the carbon atoms. These are found in natural fats called butyric acid, palmitic acid and stearic acid. Sometimes you can see all these in the that you can observe in your beauty compounds. Actually, when you will see the ingredients which are presented in the beauty compounds, you can see the presence of butyric acid, palmitic acid and stearic acid. And in case of unsaturated fatty acid, yes, some of the carbon and carbon bonds that are not saturated, they contain the double bond. Hence, they can be reduced to the saturated fatty acid by the way of hydrogenation. Example is oleic acid, palmitoleic acid, arachidonic acid. Here the complex and compound lipids. Name itself indicating that complex. The first one is simple, name itself indicating that it's a simple one. When hydrolysis, it yields the fatty acid. When coming to the complex compound lipids, these are the esters of the fatty acid that containing not only the glycerol, but also having the phosphates and the carbohydrates, sugars are the sulfates. Here, the compound lipids containing the phosphor group or sulfur group and along with the carbohydrate group, depending upon the nature of presence of phosphate, sulfate and sugar. Here you can see the examples. Lipids that containing the phosphate group are called as phospholipids. Here the example for that one, phosphatidic acid. And lipids that containing the sugars is called as glycolipids. Those were presented, for example, like cerebrosides and gangliosides. There are other lipids, such sulfolipids, like sulfolipids, which containing the sulfate, or amino group, amino lipid, which containing the amino group. So, other one is derived lipids, substances which is derived during the hydrolysis from the any above lipids like complex lipids or the simple lipids that were known as derived lipids. They include the fatty acids and glycerols and steroids. Here the examples for the derived lipids are waxes and steroids. Here the waxes are the esters of fatty acids and the complex monohydric alcohols that instead of glycerol of the fatty acids. This is having the high melting points. They form the water resistant and protective layers. Waxes do not occur in the human beings. When coming to the steroids that are not true fats, and they are derivatives of the parent nucleus known as parhydro, parhydrocyclo pentano phenanthrene, which is the ring with the 17 carbon atoms with the 3 6 numbered rings and 1 5 number ring. For example, cholesterol, male and female sex hormones, and vitamin D, adrenal cortical hormones. What are the functions? What are the major functions of the fats? Yes, body fat, which is the reservoir of the potential chemical energy reactions. The reserved fats are already available in the uh, times of restricted nutrition for the endorganic process. Exorganic means the releasing of the ATP. Endorganic means requirement of the energy for leading the metabolic pathways, which is necessary for the maintenance of the life, that is by the body fat. And beginning and being a structural component of the membranes that helps in the permeability and cellular organization, and lipids that form a rich source of respiratory energy than the proteins and the carbohydrates, and lipids that functions as the carrier of essential components like fat soluble vitamins A, D, E, K. Lipids that functions as carriers of essential compounds like fat soluble vitamins A, D, E, K. Uh, Here, this is the GK bit. Usually in the um, competitive exams, they used to ask fat soluble vitamins A, D, E, K. And lipids form heat insulators below the skin. They protect the subcutaneous fat, which acts as an insulator against the excessive amount of the heat lost. So usually people used to feel like uh, during the winter, some people know they feel like very cold. Some people they will not feel like very cold because of the presence of the fatty tissue under their subcutaneous layer of the skin definitely makes as a protective layer against the heat loss. So definitely they feel like sweating even during the very less temperatures. And hormonal synthesis from the adrenal corticocyte, corticoids and sex hormones are synthesized from the fat derivatives. Fats provides a sulfur to many organs in the body such as heart, kidney and intestine. When coming to the lipid metabolism, here lipids are rich energy over twice as many calories per gram as either carbohydrates or the proteins. Sorry. A less important function that includes the formation of insulating membrane by then the skin, which prevent the heat loss that we already discussed. When coming to the cholesterol, yes, definitely it is a chief part of the membrane system. Triglycerides is also stored in the adipose tissue, and lipids, yes, definitely it is a very important source for the metabolism. When coming to the fatty acid synthesis, both in the plants and animals, excess carbohydrates converted into the lipids. Animal cells are unable to accomplish the reverse process, whereas plant seeds rapidly convert their fatty acid deposits to the sucrose upon the germination. After the germination, acetyl CoA is an enzyme, is a compound, a common intermediate in between the fatty acid and carbohydrate degradation. A synthesis of fatty acids occur in the cytoplasm and endoplasmic reticulum of the cell, that is chemically similar to the beta oxidation process. But with a couple of key differentiation, all the enzymes that are associated in a single enzyme complex known as fatty acid synthetase. It is the carboxylation acetyl-CoA to the malonyl-CoA in the presence of enzyme acetyl-CoA carboxylase. This is the first step reaction in the fatty acid synthesis. Here you can see the conversion of the malonyl-CoA from the acetyl-CoA. There the involvement of biotin and the carbon dioxide taken place. The hydrolysis of ATP that provides the energy for the formation of the CO2 biotin complex. In the next step, the formation of the acetyl sulfahydryl group, and here the acyl carrier protein that has the sulfhydryl group, which is having the covalent bonds to the acyl intermediates. The function of acyl carrier protein in the fatty acid biosynthesis that serves as a sort of anchor to which the acyl intermediates are attached. This process is called as transacylation. Here the acyl group is transferred from a coenzyme A to the thioester to the sulfhydryl group, acyl carrier protein. So here the acyl carrier protein formation taken place. Then the formation of acetyl CoA from the malonyl CoA. And next step is the energy forming reaction. Then it is followed by the anabolic sequence of reduction. Here the anabolism means the small formation of larger molecule from the smaller molecules. When the reduction and dehydration and the second reduction, these are reversible catabolic reactions, except that both the reduction reactions are catalyzed by the NADPH rather than by the NADH and FADH. Finally, you can see the formation of the palmitic acid from the butyryl ACP that reacts with the another molecule of malonyl. Here you can see the and releasing of the carbon dioxide taken place. The entire reaction, which is repeated many times, thus the hydrocarbon chain of the given fatty acid that synthesizes 2 carbon units at a time. The overall reaction for the synthesis of the palmitic acid looks like this. Here you can see the formation of the palmitic acid. So here the summary, you can see finally the presence of fatty acid that releases a considerable amount of the energy where the combustion of the palmitic acid taken place. When the one molecule of the palmitic acid, when the combustion of the one molecule of the palmitic acid, the releasing of 23 lakhs and 38 thousands of kilocalories of energy is released. Here, the Krebs cycle, whichever the acetyl coenzyme releases in the reaction as a fifth step, that is entered into the Krebs cycle, where the synthetic reaction taken place, the formation of triglycerols and phospholipids and the cholesterol. From that, the palmitic acid is produced. Here, you can see the process. When 1 mole of ATP used, the energy is utilized, and 8 mole of acetyl-CoA is formed. 8 into 12, that leads to 96 kilocalories of energy is released, and 7 moles of FADH2 that produces 14, and 7 moles of NADH formed that produces 21 kilocalories of energy. This is how the entire reaction looks like. When coming to the summary, yes, lipids are heterogeneous compounds which constitute the major parts of the body. And as we know, 70 kg weight man containing 10 kg of lipids. Yes, definitely, it is important source of dietary constituents. They include fats, wax. The name lipid, which given to the lipid because of their heterogeneous compounds which is insoluble in the water. The animal sources are milk, cheese, butter, egg, meat, fish. And the plant sources are sunflower, groundnut oil, ginger oil, and vanaspati. And here the body fat is a reservoir of the potential chemical energy. Here the lipids are rich in energy that over twice as many calories per the gram, either carbohydrates or the protein. Acetyl-CoA is the common intermediate of the both fatty acids and carbohydrate degradation, synthesis of the fatty acid that occurs in the cytoplasm and endoplasmic reticulum of the cell, that chemically similar to the beta oxidation process, but with a couple of key differentiation. The enzymes for the fatty acids, the catabolism are in the mitochondria. This is about the entire lipids. I'm here completing my class today. Thank you. Thank you. We'll meet in the next class.